Our next speaker is Tuan Chang from National Yang-Ming University, and he'll be speaking on GenomeCAD.
Well, I would like to thank the organizers for the invitation and also for this great chance to present our work on whole genome design. Well, when I say whole genome design, I really mean whole genome design, okay? So I'm from this, the Center for Systems and Synthetic Biology of National Yang-Ming University in Taiwan. So, well, this is a synthetic biology conference, so it's all about engineering biology. But anyone here can imagine engineering without design? Well, that would be just pure trial and error. How about design without knowing the whole picture? That would be just like blind man and elephant. Well, so getting to know why you are doing what you are doing, that's very important. Here's a quote from Albert Einstein. Once he said, if I had an hour to save the world, I would spend 59 minutes defining the problem and 1 minute finding solutions. Well, you might want to ask yourself, how much time you spend on your genetic circuit design? Well, most design here you see are still doing so-called black box designing. Well, you put a so-called genetic circuits into a host cells, all you see just your genetic circuits, not host. You don't even have this host conscious circuit design. So most current design actually ignore the crosstalk between your circuits and the host genomes. So, but what actually happened then is there are a lot of crosstalk between the host genomes and your circuit. So how to know the whole pictures? Well, if you look at a current sequencing project, there are already 862 complete sequence genome available for everyone to use. And most of the sequence genome and the genomes ongoing are microbial genomes. That's why we're actually focusing on using microbial genomes for whole genome design. So we're using those genomes, the 800 complete genomes as input data, and through our mining tools, we do all kinds of genome minings to get all the parts and rules for connecting them together to come up with this so-called GenomeCAD, whole genome design tool. Well, those all 800 genomes, they are naturally existing working models. There's no trial and error. Every genome there works perfectly. Well, if you limit yourself to iGen registry parts, well, there are only 22,000 parts for you to use. And many people are not using them, or they only use only a few of them. Only a few of them are very popular, but most of them are not very popular. But if you look at genomic parts from all the sequence genomes, you have unlimited parts to use. Well, all you need to do is just make the primers with all the cutting sites and do a PCR, or they all become standardized for you to use. For iGen project, well, most people are working on so-called transcription regulation using Lac-Oblong, lambda replacers, clonal sensing, cell-cell communication. Well, that's great. But you heard a lot of talk in this conference. Most people are working on so-called metabolik engineering. Some people even talk about genome scale metabolik engineering. How can metabolik engineering can be called genome scale if you are only looking at metabolik network? Well, if you look at those naturally existing working models, those sequence genomes, well, they have all kinds of working parts, device systems for you to use. So they can be used as modules at different levels for interchange. So we're talking about a genome engineering, not changing one gene at a time, the traditional genetic engineering here. Again, our focus is on those small genomes with many complete sequences available. So you can easily do the comparisons and find the parts and the rules among them. So we're working on bacterial archaea, mitochondria, chloroplast genomes, also viral genome and phage genomes. Well, there are even more virus genome available nowadays. If you look at the sequence of phage genome, there are 500 of them sequenced already, and virus, there are more than 2,000 available for everyone to use. But why do we need a CAD for synthetic biology? Well, we are talking about genome scale. We are talking about whole genome. You need a speed to complete whole genome design. You need efficiency to avoid waste of time and effort. You want to make everything work precisely as you design. So genome engineering is about precision design. So if you look at the genome complexity, you have to have a CAT for synthetic biology. Well, look at how complex the robot is. Well, we're making a biorobot, a genome, the whole genome at the whole genome scale. Well, this is a paper describing the principal functional categories of simple living, free living organisms like E. coli or B. subtilis. There are tons of functions involved in these cells. We can divide them into information storage, processing, serial processing, signaling, and metabolism basically. But there are still other functions, detailed functions available. Well, we can start from the bottom up, or from top down. Most people use minimal genome set for their synthetic genomes. Well, but organisms like Mycoplasma genitalium, even though it has the smallest genome, the size about 480 kilobase, but it takes 3 weeks to see the colonies. For E. coli, even though it has a 10 times bigger genome than the Mycoplasma, but it only takes overnight to get B-dim cells out of E. coli culture. So why not use top-down approach? Use naturally existing working model for your engineering. Well, we're working on this GenomeCAT, or GenomeCAT is a knowledge-based whole genome design tool solution. It's a phenotype-oriented computer-aided genome design platform. Yeah, there are two major factors for this platform. One is the knowledge base for designing genomes. Well, you have to have the knowledge, information on the functional organization of the whole, everything in the cells and their interactions within the cells. Also, you need software infrastructure to do the genome scale design process from your conception to the blueprints. So this is design flow. We have actually 12 design steps for this whole genome design. I'm going to introduce all the steps to you one by one. So look at the first step. Well, you got to know your target functions. What do you want the organism to do? What's the function you want? Well, so it's going to be custom capability for this design organism. Well, besides that, we already have this bacterial phenome database. This database is composed of 781 bacterial species currently. We have 576 phenotypes included in this database. Basically, we have the serial features, 150 phenotypes out of this category, and also metabolism, enzyme activity, resistance. Here are functional specifications for phenotypes in our database for you to design. Once you have this bacterial phenotype database, you can connect with all the public available information to connect phenotype to genotype for your design. This is a user interface for your query and selection for phenotypes that you want. Once you decide the function you want, well, then we'll come to the real design step. That's the competency design step. Well, when you think about a free living organism, the first thing you want to care about is the metabolism. Well, the cell has to survive in the environment. We have this tool called part of this GenomeCAT. So you can go through all the 130-something metabolic pathway to design your genomes. So everything is in the metabolism. It's included in here. So not only bacteria, also include eukaryotes. For this competency design, well, on the top here you have 131 reference pathway for different functions of metabolisms. For each horizontal line here, present the profile, metabolic profile for the organism you're interested in. Therefore, you can compare hundreds, thousands of genomes all together at the same time to see the similarity and differences. The blue color means they are very similar to each other. The red color stands for differences. So once you do a comparison, you will know the metabolic profile of all organisms available. So you can do a pathway clustering to see which organisms have similar metabolic profile. Then you can look at the metabolism activities to see where you want to start with those genomes for your design. See where their functions in terms of metabolism fit with your target functions. Or you can do pathway sorting from the dissimilarity to very similar organisms. Once you have the metabolism function design, then you will come to this composition design function. Well, we actually have our own pipeline for composition design. That means everything in the genome Once the genome is sequenced, all the genes are annotated also, but we have our own pipeline to annotate, to do gene prediction annotation for all the sequenced genomes. Why? Because if you look at all the genomes out there at NCBI, Gene Bank, they are done by different groups. But different groups use different criteria, different pipeline, different tools for gene prediction annotation. How are you going to do comparison mining if they annotate differently? So that's why we have our own annotation pipeline. We call it the Bacterial Genome Annotation System, BGAS. So we use the same tool, same pipeline, same process to annotate everything available. So here's just to show you all the genes. We annotate the functional categories, browsers to browse through the genomes. So you can choose the genome you want to see according to your function. But there's still a problem. Even though genes are annotated, once the genome is annotated, you still have these genes with unknown functions, so-called unknown genes. Then how can you deal with this unknown gene for your whole genome design? Well, we have this tool that can actually help you out with the unknown function in the genomes to see whether this enzyme, this gene is really missing or is there. So this is a user interface for our composition design to finding unknown genes, so you can do a comparison across genomes. They can come up with the chromosome location comparison with their functional pathways. So you can see here we have 5 chromosome location from 6 different species to make a so-called conservation profile. Then you can use this profile to search against your target organisms to see where those genes, those enzymes are really there or not, and map them to the pathway to see the correspondence. Also, we have another tool to rank those hypothetical proteins. We call it HyperRank. So you have all kinds of criteria for you to compare those unknown proteins to see which one, if you want to delete them or keep them, and how you can select genes to keep or to delete. So we have this hypothetical protein database to rank all the hypothetical protein for functional design. Here are the criteria we use for ranking them. Then we even have a search interface for you to search if you don't want to do your own annotation, unknown protein design or search, then we can do it for you. And once you have a composition, everything besides the metabolism there, then you want to go with this replication design. Because for synthetic whole genome, you want the genome to be able to replicate from generation to generation. Then you have to worry about you're going to do a single replicon in the cells with a single ORI, or you can do so-called quad-code, dual-code quad-code genomes, like Vibrio has 2 chromosomes in the cells. And then you have to worry about the compatibility problem, the genes along this origin replication, and also the copy numbers. So we have this tool for you to compare the origin replication, the genes around this origin replication, how the range, what's the rule for design, putting genes around the origin replication. We have this ORI design tool, or you can map out the DNA box and the DNA binding sites. Also their skew, optical skew properties. So this is our online prediction service. So you can put in your sequence, we can predict the origin replication for you. Also, we have a pre-computed database available for all the sequence gene already. So the replication is designed. Then what? Then you have to put the genes all together. Not individual. Well, genes in bacteria, they don't exist individually. They form operons. Or some of them form so-called monocitronic operons, but most of them form so-called polycitronic operons. Then how can you put them together? What are the rules for putting genes together? How about gene context? Which gene I can put with other genes? How about operon structures, transcript units? So we have a pipeline to do operon predictions to using different criteria for the cutoff to find monocitronic genes and polycitronic genes in the bacterial genomes. So you end up with a figure like this. This is a different function, called functional category for different genomes. So those are functional categories. Then you can use low threshold, low cutoff, or high cutoff for your operon predictions to see what genes you want them to be put together, become polycitronic operon. What genes you want to put them into monocitronic genes. So That's a design tool from Alpine studies. So you can even do cross-genome comparison to see the rules from multiple genomes. Once the operons are formed, then you want to put everything onto the chromosomes. A global design, global genome organization. You have to worry about how you can put the gene. You can put gene on top, bottom, on the side. What strand you can put gene? On top strand or bottom strand? Where you can put gene? You can put the gene in front of another gene or after another gene. What's the rule? So we have the rule for whole genome comparison organizations. So we have tools. You can, well, this is a Java tool. Actually, you can compare all different genomes in terms of different genomic features. So this user can do all kinds of selection, but I don't have time to go to the details. So this is the result. So once you select the feature you want, then the tool will automatically compare all the features for you. So you can compare one genome, different features, one genome. You can put different genomes all together for comparisons. Well, this tool has many, many different functions. I don't have time to go through that. I would just rush through it. But this is not only for circular bacterial genomes. It can also be used for linear genome like human, mouse, linear chromosomes. Here's the example to show you the comparison results for the rules. Well, so once you found how you can put the gene back to the whole chromosomes, then you have to worry about the regulation design. Well, you don't want to just put gene onto chromosome. What's the relationship between the genes? What are the regulation networks? Well, we actually did the promoter comparison for all the operons in the genomes. So you can compare upstream regulated sequence of all kinds of bacterial operons to see whether they are similar to each other. So, for example, here you can compare different strains within the same species. So often Bacillus subtilis, but they come with different strains. What's the difference between them in terms of promoter sequences? How about different species within the same genus? Is there a difference? Different genera within the same families. You can go higher and higher to see the design rule. It's different labels, different families within the same orders. You can even compare the promoter sequence, the operon promoter sequence between gram-negative and gram-positive bacteria. So there are all kinds of rules existing in the nature's working models. Also, for the regulation, you want to know the transcription regulation elements in front of your open friend or after your open friend, the transcription factor binding site and terminators. So we do all kinds of motif binding by using in silico bioinformatic process. We get data from RegronDB for transcription factor binding site. We get terminator information from RegronDB to compare for our pattern binding. We put elements back to the promoter to see architecture, to see how they similar to each other in terms of whole genome structures. Then we can also use the experimental data to put the regulation network back to your genomes. So this is an example to show you. We actually did the phosphate sensing in E. coli experiment with our collaborators. So we using chip experiment. We should using chip sequencing experiment to find all the binding sites. So you can see the 4B binding site in the whole genomes. And you can map them to the gene orientation functional categories. Then you will end up with a regulation network starting from the signal of phosphate starvation. Then the signal will come in, connect with the sequence transduction to transcription factors to downstream metabolisms. So then this is just a single modules in the cells, but there's still a lot of other transcription modules in the cell too. I'll show you later on. So rules from natural existing working models coming from the signal from outside into the receptor on the surface of cells and going to the cells with the transcription factor response elements and with the regular gene and downstream metabolisms. So you have everything connected all together. And you can map all the experimental data or predict the pattern motifs to all the genomes to see how they're located throughout the whole genomes. You can connect the motif binding site with the functional category downstream genes to see the rules. Then you can map all the known transcription factor experimental data to downstream function 2 to put them together, prediction and experimental data. So you have a transcription replication network. But bacteria also have this so-called small RNA regulations. We have a tool for small RNA regulation prediction. So this is the small world of bacteria. So we can predict, and also using experimental data, to find all the small RNA or ribose switch existing in the current cells and put them back to the regulation network to see how the signal are transduced from transcript factor to other genes, how the genes are translational regulated by small RNA and ribose switch. Once you have the regulation design done, then you will go to the next one. There's a whole cell interaction network design. Well, cells has all kinds of functions. For example, signal transduction. Well, cells have to sense what pH in the environment, oxygen, nutrients, all kinds of signal from the outside. So there, for example, in E. coli, there are two component systems. Proteins are responsible for transducing those signals. So we have signal transduction figured out by using literature experimental data too. We have transport system mapped already. We have gene regulation by transcription regulation network established. As I just mentioned, we have this small RNA ribose switch gene regulation and transcription label. We have MedTops network mapped through this KGG reference pathways. We are using protein-protein interaction data from experimental results. So put them all together, we have a single network. We don't have separated network. We're not only working on metabolism, we're working on everything in the cells, single network. So by connecting them all together, we have a whole interactome in the cells. So this is an example for E. coli. E. coli has about 4,000 something genes. Well, exactly, we have 4,000 everything in there. We have all the connections. We have 25,000 connections among them. Then we can have all kinds of assistant label property to map out all the hubs and put all the essential genes back into the network. Not only so, we also put gene expression experimental data to make the whole network dynamic. So now you have quantitative data. You can use them for temporal and spatial rules. So you know what genes will be turned on at what time, under what conditions. So we have this bacterial gene expression data composed of transcriptome, proteome, and metabolomes. All those are gene expression data from experimental results. So this is the current database condition. So this example to show you, for example, this is a published paper in year 2006 on this BMC microbiology about core pH dependent genes. So you can see what genes are regulated under what conditions, aerobic and anaerobic. You can see genes regulated in the presence of different carbon source. This is going to look like just like a movie, actually dynamic moving in the cell. So you start with glycerol and change to cysteine, alanine, acetate. Well, you are looking at a dynamic picture for the whole cells. Genes being turned on, turned off at different conditions, different times. The whole network is dynamic, not static. Well, we have this genome cat. You can explore the whole genome through our query interface, through all the sections, search, design. But, well, depends where you want to do epigenetic design. Because most bacteria, they actually have this restriction modification system implemented in their genome already. So maybe you want to put the different types of restriction modification system in the genomes and different numbers of them, how to balance them for the defense. Well, once you go through that, it will be design verification. You want to verify everything designed in the gene for the whole genome. From the two-component system, we have two-component system mapped already. Everything for the E. coli. So you can connect them from outside to inside, down to everything. And we also have the proteome mapped already. You can see how the protein are composed of the domains. See the protein domain combination graphs, natural existing rules there. You can do all kinds of domain swap for your engineering purposes. Then you can do cross-species comparison to map the signal transduction to other organisms, other species, to do the whole genome design. And last step will be design for manufacturing. Well, once you finish the design, you still need to think about how you can manufacture the whole genome. So then you have to worry about the step, which part you can do the synthesis first, how to do the implementation. You have to check the physical property of the whole chromosome, the curvature, the bending, the optical skew, everything. Then will be the last step for genome synthesis. Well, from this conference, you know a lot of industrial company and academic research group are doing whole genome synthesis. We are not doing that. So a lot of expertise they are there. You can send out for them to do your whole genome synthesis. We're also working on lactic acid bacteria. We are working on pathogenic bacteria for the same thing, for the whole genome design. Also phage genome too. So you can connect experience data to your whole genome information. So this is my summary. So we have developed a knowledge-based genome CAD design environment for computer-aided whole genome design of microorganisms from phenome to genomes. So it's an integrated tool, composed of a lot of components. We also have this temporal, spatial, dynamic features implemented with experimental data. We have this can be used for, well, GenomeCAD can be used for both academic biological mechanism exploration, also for industrial application too. Well, without them, without all the hard workers out there in my lab, you will not see GenomeCAD today. So there are many people involved in this project. So they did all the excellent work for this project. So I will end this talk by thanks to the financial support from National Council, Science Council from Taiwan, also some of our collaborators from biotech, pharmaceutical, information technology company. I'll stop here. Thank you.
So we probably have time for one quick question.
As I just mentioned, well, this knowledge-based, well, the design tool, it's just software tool, but the knowledge-based, this is an important part. But the knowledge-based containing a lot of proprietary information data and our collaboration data. So we are still thinking about how to release this design environment to the public. Yeah, well, you can email me. We can talk about this later on.
All right, let's thank our speaker again.
Thank you.